Assalamualaikum and hi everyone. Let's continue the second part of the pharmacodynamics, which is drugs actions. So as we discussed earlier, the target for the ligands or the drugs are mainly on the receptors, ion channels, transporter and enzymes. So today we will focus on this target protein. So let's continue with the receptors. So, receptor has high and selective affinity for the drug molecule that elicit the biological response. Agonist is a drug that binds to the receptor and activates it. It can be a direct action or through the second messenger, or we call it as a transduction mechanism. The antagonist is a drug that occupies the receptor but do not activate them. So there are lists of the receptors that are available in our body system. As example, acetylcholine receptors, it can produce the effect by binding with the acetylcholine. The tuberculin can block the acetylcholine receptor being activated. So I want to highlight here the histamine and loratadine. The binding of histamine on the H1 receptor will produce symptoms of sneezing, itching, watery eyes, and runny nose. The binding of the drugs, which is loratadine, will stop all these symptoms. Let's have a look.
Histamine, in turn, binds to H1 receptors on the surface of cells in different areas of the body: eyes, throat, nose, stomach, skin, or lungs. Histamine binding to the H1 receptor sends a signal to the inside of the cells, in turn causing fluid leakage, mucus production, or other responses in an attempt by the body to rid itself of the offending allergen. Loratadine and other antihistamines bind to the H1 receptors that exist on the surface of many types of cells. This prevents binding of histamine to the H1 receptors and shuts off the signal cascade that leads to allergy symptoms. Loratadine and desloratadine are effective because they bind to the H1 receptors tightly, stay active in the body for several hours, and do not generally cause drowsiness.
So let's move to the second part of the target protein, which is ion channels. Drugs can affect ion channel function by interacting either with the receptor site of ligand gated channel or with other parts of the channel molecule. The ion channel blocker or the drug can block the ion permeations, while the modulator might increasing or decreasing the opening probability of the channels. So I want to discuss about the amlodipine, which is an antihypertensive drug that causes vasodilations. Amlodipine acts on the calcium channel by blocking the calcium channel. Calcium channel can be found on the cell that lining along the blood vessel. The binding of the drugs, which is amlodipine, on the calcium channel will block the influx of calcium ion. Let's have a look on the video attached.
These blood vessels are lined with cells, and these cells contain channels called calcium channels. Your body tends to have a lot of calcium outside the cell, represented as Ca+ here. When signaled, the calcium enters the channel and goes into the cell. This tells the body to make the blood vessel more narrow. When blood needs to go through a smaller space, the pressure increases. Thus, it increases your blood pressure. So how do calcium channel blockers work? Amlodipine, a calcium channel blocker, will enter your system and block the channel. When this happens, calcium is unable to enter the cell. As a result, The blood vessel cannot narrow. In fact, it can cause vasodilation, which means the blood vessel can widen. Now that the blood has a bigger space to go through, the pressure decreases, leading to lower blood pressure.
So let's move to the third part of target protein, which is transporters. The transport of ion, such as natrium, calcium, and small molecule, which is glucose and amino acid, across these cell membranes, generally requires a carrier protein. The drug may act as inhibitor, blocking the transport system. So this is an example of the transporter and also the drugs inhibit. So I attach here the video of the mechanism of action of omeprazole that inhibit the proton pump from secrete the gastric acid. Let's have a look.
Acid is produced in the stomach to help break down food that is eaten. It is produced by a proton pump which is found in the lining of the stomach. Sometimes the contents of the stomach flow back up into the food pipe, also called the esophagus, all the way up into the throat, which can cause symptoms such as a cough, hoarseness, and voice problems. This is called laryngopharyngeal reflux, or LPR. To help with the symptoms of LPR, doctors may prescribe tablets or capsules from a group of medicines called proton pump inhibitors, or PPIs. There are a number of different PPIs, for example, omeprazole, lansoprazole, pantoprazole, rabeprazole, and esomeprazole. They all work by blocking the proton pump to reduce the amount of acid in the stomach. As a result, they can reduce acid reflux, which may help relieve the symptoms of LPR.
So let's continue the last part of the target protein, which is enzyme. The drug molecule may act as substrate analog for substrate as well as prodrug. For the substrate analog, they act as a competitive inhibitor of the enzyme, whereas they are reversible or irreversible bound to the enzyme. The fourth substrate, where the drug molecule undergoes chemical transformation to form abnormal product that subvert the normal metabolic pathway. Whilst the prodrug, which is inactive form that require enzymatic degradation to convert to an active form of the drugs. So there's an example of the enzyme and the drug that inhibit the enzyme. So I want to highlight here about the simvastatin. Simvastatin is a lipid-lowering drug, or also known as HMG-CoA reductase inhibitors, that prevent the synthesis of the cholesterol by inhibiting the cholesterol synthesis pathway.
The cholesterol synthesis pathway involves multiple steps. The most important step of the entire pathway is the formation of mevalonate from a molecule called HMG-CoA. HMG-CoA contains coenzyme A, which is removed to form mevalonate. This reaction is carried out by an enzyme called HMG-CoA reductase. It turns out that statins can also bind to this enzyme because statins and HMG-CoA have similar structures. However, HMG-CoA cannot bind to the enzyme if a statin is occupying that binding site, meaning that no reaction occurs and no mevalonate can be formed. This is how statins stop cholesterol synthesis.
Let's continue the last part of this lecture, which is signal transduction. Ligand, or also known as primary messenger, will act as a signal. When these ligands bind to the receptor, which is act as a signal detector, will induce the secondary messenger that produced within the target cell. These secondary messengers relay signal from one location to another, leading to cascade change. So this process also known as signal transduction, or also known as cell signaling, which is transmission of molecular signal to produce the response. So I want to highlight here, drug can be act as a signal and the receptor acts as a signal detector.
Let's talk about the general sequence of cell signaling. First, reception. Typically, a signal molecule binds a receptor. Second, transduction. The receptor gets activated by this binding. This often means the receptor will change its shape. It could even involve a whole series of molecules changing their conformation in something called a signal transduction pathway. This can amplify the original signal. Third, response. There's some kind of response that is going to happen. A portion of DNA that is found in the nucleus getting transcribed, for example, that's a type of response. Now, cell signaling can involve intracellular signaling, which occurs within the cell itself, and intercellular signaling, where a cell communicates with another cell. In many cases, signaling involves both, signaling between cells and then also signaling within cells. Now, when we're talking about signals traveling from one cell to another, distance matters. Some cells are close and have direct contact. In the case of these two animal cells, they are gap junction close. Gap junctions in these animal cells, or plasmodesmata in plant cells, are connections between two close together cells that allow ions or other small molecules to pass, and by doing so, they don't have to pass across the cell membrane. Paracrine signaling allows a cell to target another cell by a signal molecule that may diffuse between them. These cells are still close but need not be connected. The ligands in paracrine signaling tend to be rapidly absorbed and rapidly degraded. The ligands are typically not traveling far, as this is local signaling. Synaptic signaling, which specifically involves neurotransmitters in a synapse, is another example of this local signaling. And what about long distance? Endocrine signaling can allow a cell to communicate with a target cell from far away. Signals may be carried in the bloodstream. Hormones released by certain types of endocrine cells are a great example. We also want to point out that a cell could just signal itself. For example, in autocrine signaling, a cell could secrete a certain type of molecule which then binds to its own receptor and causes a response. A cell releasing its own growth factor could be an example of this.
So there are two characteristics of signal transduction, which are signal amplifications or desensitization and downregulation of the receptors. Signal amplification is the ability to amplify the small signals based on the duration and intensity. As example, liver cells can convert the glucagon into the glucose by detecting just a single molecule such as epinephrine. And the second part, which is desensitization and downregulation of receptors. The responsiveness of the receptors has been declined with time, which will cause the lower the effect or efficacy of the drugs due to the repeated or continuous administration of the drugs. It is important to protect the cells from excessive stimulation and prevent any damage of the cells. There are two types of desensitizations, which are tachyphylaxis and tolerance. The difference between the tachyphylaxis and tolerance can be seen by their time to onset the actions. The decrease in the drug effect for tachyphylaxis occurs rapidly, whereas the tolerance, which took days up to few weeks. So that's all for the pharmacodynamic part of the lecture. If you have any problem, if you have any question, please discuss on the forums down there. Thank you everyone. Keep calm, stay safe. Assalamualaikum warahmatullahi wabarakatuh.